Hi everyone, my name is Zishuo Zeng. I'm from Dr. Yana Bromberg's lab at Rutgers University. Today my topic is a novel framework to evaluate the effects of synonymous variants. Synonymous variants are the nucleotide variants in the coding sequence that do not change the protein product. For this reason, many people may overlook their effects. In fact, they can affect biological functions in many ways, including transcription, splicing, stability of the mRNA, co-translational folding, tRNA wobble, etc. Therefore, they can potentially lead to disease. There are currently 9 predictors. We reviewed them and found that they may suffer from the following limitations. First, the reliability and size of the data are compromised. Second, most of the predictors account allele frequencies for effects, and the performance of these predictors are often conservation driven. So to build our own novel predictor, we want to increase the size of the data. We don't want to filter the data by the allele frequencies, and we don't want to incorporate the conservations into the feature set. So how do we get the bigger and better data? So we know that there's a relatively large sequencing cohort called GLOMAT-ASOM, and from which we extracted about 4.1 millions of observed synonymous variants that are mapped to the transcript level. Besides that, we have also 4 millions of singletons, which are the variants that are observed in only one individual. For our modeling, we do not incorporate the singletons into the picture. And apart from these two, we artificially generated about 57 millions of the synonymous possible but not observed synonymous variants. And how do we make use of this data? So let's say we plot the observed and generated variants into a two-feature space. The blue ones are the observed, and the yellow ones are generated. Some yellow ones are close to the observed ones. We call them not seen. They are either observable if we have more sequencing samples, or they are deleterious and have been purified. And the remaining ones are in the corner that they are very different from the observed ones. We call them as unobservable. We may never find them because of the constraints of our model, our features, or because of the physiochemical constraints of the DNA structure, or even the technical constraints during the sequencing process. So the very first step for us is to separate the generated variants into the not seen and the unobservable. To do that, we use positive unlabeled learning. In our case, the positive data is the observed, and the unlabeled data is the generated. So basically, we sampled a part of the equal amount of generated as the observed, and then use it to train a machine learning model. The machine learning algorithm we use is extreme gradient boosting, and we use this model to score the generated variants, and then the ones that are predicted as observed or positive, they are taken out and added to the not seen group. The remaining ones, we use it for further training, like starting a new epoch for the new generated set. We repeat this process until the PUL process is converged. By doing this, we have conducted about more than 100 epochs, and we determined that the PUL process converges at epoch 63, giving us 51 millions of the not seen and 6 millions of the unobservable. So we train a model using the observed and not seen. The purpose of this is to identify the observable in the not seen. So this is the result. From the not seen, we can say that in the future we may observe maybe 10% of this not seen group, which is about 4 or 5 million in the future. But at this point, the model is only differentiating observed and not seen. So what we want actually is a model that differentiates effects. So we want to further subset the data to build a model for that purpose. For this purpose, we bring common variants and pathogenic variants into the picture. Common variants are the variants have allele frequencies greater than 1%. They have little deleterious effects, otherwise it wouldn't become common. Pathogenic variants are those that are experimentally verified to be disease-causing. So pathogenic variants are observed, but they are predicted high up. The reason for that is because they are similar to the purified ones in the not seen group, so that we believe that the high score not seen groups is enriched for deleterious is enriched for effects variance. On the other hand, the common variance is very similar to the observed ones, and we believe that is reflected of the neutrality of the observed ones. So when we subset data for the no effect group, we want to keep it below 0.5 to be consistent with the model, and we also want to retain the majority of the common variance. So we can do it this way. We subset the not seen group up at a line that above 75 percentile of the pathogenic, and then we subset the observed group below the 25 percentile of the common variance, and then we use these two subset data to train the final model. This is the final model predictions for no effect and effect groups in the testing set. We call our model SYMVAP, synonymous variance effect predictor. And then we also determine a new cutoff for our final model. Basically, we took the mean of the medians of commons and medians of the pathogenic, and then using this cutoff, it gives pretty good precision and recall. And we also evaluated singletons using the final models. We found that although most of the singletons are predicted below the cutoff, it is still slightly higher than the observed ones. We also compare our predictor with other predictors. We found that all of the predictors can pick up the signals between the testing set no effect group and the pathogenic group. But we also found that for all the predictors, the two groups either both fall above the cutoff or below the cutoff. Only our predictor, the two groups falls on both sides of the cutoff. That's all for my presentation. Thank you very much.